Alright, we're going to continue our lecture on structure and function of large biomolecules, lipids, and nucleic acids. The characteristics of lipids begin with the fact that they are not true polymers. There is no true polymer formation. They are relatively small molecules, and they do not fit our definition of macromolecule. Lipids are also hydrophobic and highly nonpolar, with lots of carbon and hydrogen bonds in their structure. Last, there are three main groups of lipids that we are going to look at. One is triglycerides, followed by phospholipids, and then waxes and steroids. Starting with the triglycerides, they are made up of 3 fatty acid chains and 1 glycerol molecule. This is a sugar, it is a triose, and otherwise known as an alcohol because of the hydroxyl groups on the structure. Each fatty acid is going to be attached through dehydration synthesis to form what we call ester linkages. Ester linkages are circled in blue. Water was removed, a hydroxyl group from the fatty acid, a hydrogen from the glycerol. There are 2 types of fatty acids, unsaturated and saturated. We will look at both types. The functions of fats are that they have greater energy storage. They are 2 times greater than polysaccharides, starches, and glycogen. You can combine more energy in a smaller molecule. Fatty layer in animals protects and cushions organs. This is the purpose of adipose tissue. Animals move around. We have to carry our energy stores with us, so we developed adipose tissue. It swells and shrinks as needed. It is also an insulating layer in marine mammals. This insulating layer helps keep these marine animals warm in very cold water. Moving on to saturated fats. These are a very linear structure. Because these are linear, the molecules can stick together easy, thus forming a solid at room temperature. Butter and lard are examples of saturated fatty acids, and they're usually made up of animal fats. Unsaturated fatty acids, on the other hand, have a double bond between the carbons of the fatty acid chain. You can have one double bond, which would make it a monounsaturated fat, or you can have many double bonds, which would make it a polyunsaturated fat. These are usually going to be found in plant seeds and fish, and are commonly called oils because they are liquid at room temperature. Naturally, they are going to be found in the cis configuration, where your hydrogens, or the groups coming off of the carbons in the double bond, come off of the same side. This is going to form a bend in the molecule. Trans fats, on the other hand, are fats where you still have a double bond, however the hydrogens are on opposite sides. This is due to the hydrogenation process, where naturally occurring cis fats are hydrogenated, and part of this process is to make them unsaturated. Part of this process is to make them saturated, and also we get trans fats out of this. It is known that trans fats contributes more to cardiovascular disease than regular, normal saturated fats. Hence the health issue with trans fats being banned in certain areas or regulated. Food labels have to be labeled with the amount of trans fats or hydrogenated fats on the package. Steroids and phospholipids are another group. Steroids are made up of 4 fused carbon rings. Examples of steroids are cholesterol and cortisol. These maintain proper membrane structure in animals, and they are also the precursor to steroid hormones such as estradiol and testosterone. Phospholipids are similar to that of triglycerides, except it has two fatty acid chains, a glycerol, and a phosphate group attached instead of a third triglyceride. So glycerol, two fatty acids, and a phosphate. These are going to be key components of cell membranes. Moving on to nucleic acids, we have the monomer is a nucleotide. It is a 5-carbon sugar with nitrogen base on carbon number 1, phosphate group on carbon number 5, and phosphodiester bonds will form by dehydration synthesis between the phosphate of one nucleotide and carbon number 3 on the second. Nitrogen bases can be of two types. You have pyrimidines and purines, single ring or double ring. The way I remember it, a pyrimidine has a long name, pyrimidine, and shorter structure, while the purine has a short name and long structure. You can also remember pure silver. The purines are made up of adenine and guanine, pure AG. That's how I remember adenine and guanine are both purines, by pure silver. The structure of nucleic acids is very, very similar, but there are differences. The two main nucleic acids are DNA and RNA. The sugar is deoxyribose in DNA and ribose in RNA. On carbon number 2, there is an oxygen on ribose to form a hydroxyl. In deoxyribose, it's only a hydrogen. DNA is made up of 2 polynucleotides, whereas RNA is made up of a single nucleotide strand. A nitrogen-based thymine is found in DNA, but not in RNA. DNA stores and copies information, while RNA transmits information and performs other functions as well. Let's look at these in a little bit more detail. Nucleic acids, DNA, otherwise known as deoxyribonucleic acid. These are polymers joined by dehydration synthesis to form a phosphodiester bond. This bond is circled in red. It's a phosphodiester because you have a phosphate group followed by 2 oxygens in the bond, linking the 2 nucleotides together. So 2 esters instead of 1. Hydrogen bonding occurs between the 2 polynucleotides. Base pairing always follows adenine nitrogen base with thymine nitrogen base, guanine with cytosine. It also allows for DNA to have a specific direction. Notice the arrangement of one side of the nucleotide strand versus the other side of the nucleotide strand. They go in opposite directions. So we have complementary base pairing and opposite direction. So ribonucleic acid, on the other hand, is very similar to DNA, with just a difference in the 5-carbon sugar and the difference in the 1 nitrogen base. You still have polymers formed by dehydration synthesis. It's still joined between phosphate of 1 nucleotide and the 3' carbon of another nucleotide. And even though it's single-stranded, it can still fold over on itself like in tRNA or ribosomal RNA. There are several types of RNA that do different functions. Messenger RNA is going to carry information from the nucleus to the ribosome. Transfer RNA is going to carry amino acids from the cytoplasm to the ribosome for protein synthesis. And ribosomal RNA is actually going to be part of the ribosome and help with the process of joining the amino acids to form the peptide chain. Another type of RNA plays a role in gene regulation. We will talk about this a little bit later on in a different course. But this is non-coding. It does not produce a protein. It's strictly for gene regulation. The last segment I want to talk about is specialized nucleotides. They don't have to be in a polymer in order to be important to a biological system. Adenosine triphosphate, otherwise known as ATP, is a very, very important nucleotide. It is a nucleotide triphosphate. There are others. Each of the 4 nitrogen bases form a trinucleotide. But this one is unique because it is going to be used for cellular energy. It is quick energy. It's used as quickly as it's made. It is not long-term energy storage. We'll become familiar with this in energetics. Other modified nucleotides include the electron carriers that we'll also talk about in cellular energetics, known as NAD and FAD. This concludes the lesson on lipids and nucleic acids, and I will see you again in proteins.